Hello and good afternoon. My name is Sharon Peacock and I'm the Director of the COVID-19 Genomics UK Consortium, or COG-UK. So I'd like to extend a very warm welcome to you and thank you for joining us this afternoon at our latest COG-UK Science Showcase. So during the next 2.5 hours, you will hear a series of talks focused on how the sequencing of SARS-CoV-2 performed by COG has contributed to core national studies in the UK. So we've been working closely and collaboratively with everyone that you will hear speaking today, and I want to extend my grateful thanks to them for their willingness to work with us at COG and for their spirit of collaboration and teamwork, and also for doing the incredibly important studies that they have that's delivered towards the pandemic response. So turning to our programme today, we've divided the meeting into 3 sections. The first will look at the use of sequencing during hospital-based studies and vaccine studies. This will include viral genomics to better understand hospital onset COVID-19 and contributions to the interpretation of vaccine efficacy. We're then going to move straight on without a break into our second session, looking at the impact of genomics in a range of different settings, including care homes and universities. We'll then take a short break before going on to our final session, which will cover sequencing for public health and national infection surveys, including the study run by the Office for National Statistics, or ONS, and the REACT studies. So before we get started, some very brief housekeeping notes. Please note that the session is being recorded and will be posted on our YouTube page with links from the COG-UK website. For everyone not speaking, please remain on mute and with your camera turned off. Each talk will run for 15 minutes, which includes time for questions. The chairs will aim to keep the meeting running to time, and so we will only take questions at the end of each talk where time allows. There's a Q&A function for your questions, so please do keep your questions coming, and we will forward these on to the speakers as we go along. So without further ado, I would like to hand over to our first chair, Dr. Thushan de Silva, and we'll get started with session 1. Thank you very much.
Thank you, Sharon. Welcome to session 1. As Sharon mentioned, we have some great talks for you today in this first session, focusing both on the role of sequencing in hospital-acquired infections as well as in the vaccine efficacy studies. So it gives me great pleasure to introduce the first speaker, who is Professor Judith Breuer from Imperial College London, who's going to speak to you today on the role of viral genomics in the COG-HOCI study. Over to you, Judy. Thanks.
Sorry, can you hear me now?
Yes, we can. Yep.
And can you see my screen?
We can now, Judy. Yep.
I put it on. It's not on. Okay. See, you can see the first slide.
That's great. Yep.
Great. Okay. Good. Thanks very much for this invitation. I'm going to just briefly whistle-stop through the COG-UK HOCI study. The rationale for this was that many of us are involved in this area, and we know that sequencing pathogens has been many times shown to better identify the sources of infections and transmission routes than infection prevention alone, infection prevention control alone, but it's always been retrospective. And some nice work done during the first wave of SARS-CoV-2 reported that sequencing the virus improved management, but didn't really quantify. So our aim was to try and quantify this, and it was a phase 3 prospective interventional cohort superiority study. It was supported by urgent public health funding and support. We asked 2 questions. Does the availability of SARS-CoV-2 sequence data to infection prevention and control teams reduce the incidence of hospital-acquired SARS-CoV-2 infection and reduce the number and sizes of hospital outbreaks? And is the impact of sequencing on IPC practice dependent on time to result? So the methods were that we had 14 sites which were distributed across the country, 1 in Scotland and the other 13 in England. They sequenced all SARS-CoV-2 positive patients and healthcare workers from their site. And overall, this amounted to over 30,000 patients, and we aimed for 2,000 hospital onset COVID infections. Sequence data, patient metadata, and community sequences were integrated into a probability model to try and identify hospital COVID infections and to identify linked infections and report this in a simple format. The baseline ID data was compared with with rapid turnaround time, 48 hours, and a slow turnaround time to receiving the reports. And we had qualitative evaluation of why it worked or did not, and a health economic study. So just to leap straight into the genomics, there are many things we could talk about from this study, but I'm going to focus on the genomics. This is what we were faced with using at the start of the study. This was the reports that were being issued by COG-UK, which showed linked infections. And you can see that in this case, it's very hard to determine which infections are linked, because many of the infections are identical. And you would have to try and work out from this phylogenetic tree with which ones were linked together. This sort of data was then had been used by the Cambridge group with a lot of very nice addition of metadata. But you can see again, this is incredibly difficult to interpret by people who are not experts in phylogenetic analysis. And the reason that SARS-CoV-2 genomics is so difficult to interpret for hospital transmissions is because the mutation rate is low. And so you do not reliably get a change in the a difference between viruses that are not related, and you can erroneously think that identical viruses are related when in fact they just happen to be circulating at the same time. So to try and overcome this, we developed what we call the sequence reporting tool, and this compared the sequence from a HOCI case, somebody who was PCR positive or symptomatic 48 hours or more after admission, with sequences from all the other positive cases in the hospital. So we sequenced everything in the hospital. We compared that, and this is where the COG-UK consortium really came into its own. We had really good data on community viruses. We were able to look at the viruses with postcode, not only from the community, but also from the hospitalized patients. And then we were able to look at data on where the hospitalized patients were and their dates of admission. We defined identical sequences being less than or equal to 2 SNPs difference. That was just an arbitrary thing, but based on the data that came out of the initial hospital sequencing studies. And then the model used Bayes' theorem to assign a probability that a new HOCI is hospital acquired and identified putatively linked cases and where in the hospital they were located. So a complex model, but very simple initially because we had to get on with the study. We issued a simple one-page report which had the details of the patient, the likelihood based on the model, the probability model that the patient had acquired the infection in hospital and in this case on the ward, and the list of patients who might be part of the same outbreak, and then a schematic to show where they fell in relation to a high-risk period. This is the index case, and this is the potential cases that might have transmitted. We tested the sequence reporting tool in a retrospective data from Sheffield and Glasgow, and we showed that it did confirm 91% of the probable and definite HOCIs, and the likely linked cases in 95% of those. And it identified the high risk of nosocomial transmission and linked cases in about 1/5 of those indeterminate cases, with 70% of people who indeterminate HOCIs being identified as not likely to have been acquired in hospital. So potentially useful. The recruitment for this study was excellent. We recruited over 2,000 people. The sites were absolutely amazing in their ability to try and recruit these patients. Mean age was over 70 years of age, mostly white British. Most people were asymptomatic, and most people did have a comorbidity, and most of them came in through the emergency room. On analysing the results, it became apparent that 7 of the 14 sites had major difficulty in delivering this study. This was a new study, never been done before. No IPC studies have been done in this way and delivered in the middle of a pandemic. It's not surprising that 7 of the sites could not really deliver the results. And that's in itself interesting, because the reasons for not being able to deliver it will inform potential future studies as well. So the reasons were related to probably the number of HOCIs. The more HOCIs you had, the more community transmission, the more difficult it was to deliver this study. Links between the diagnostic sequencing lab of those who were not really able to deliver the study, all of them were distant from their sequencing lab, whereas 3 out of 7 who did it successfully were actually embedded within the diagnostic laboratory. And prior experience of using sequencing for managing outbreaks did seem to have some effect, but less than one might have imagined on how well the sites did. So did it have an impact on IPC activity? Just looking at those people who we deemed as able to deliver the study, you can see that they delivered the majority of the sequence reports within the study period. The mean turnaround time within the slow period was 13 days, and within the fast period was 4 days, and that's from sample to sequence. So that's an amazing result. The overall impact on IPC activity was low in the slow, was low in the fast turnaround time, but higher. 10% said that there was a change in IPC activity, and that varied by ward, more so for people on the same ward, and slightly less for people on a different ward. So it did change practice in 10% of cases, which I think is amazing in the middle of this pandemic. It was useful in 79% of cases in the fast turnaround, but not so much in the slow turnaround, where it was actually only 27% said it was useful. And bottom line, that the odds ratio of the SRT of the sequence or changing IPC practice for the fast versus the slow phases was 15.55, and that was significant. It did not identify linkages that had not previously been suspected by IPC investigation. So that's quite an exciting result. The other thing to note was that this is a pictorial representation of whether people found sequencing useful as a function of turnaround time. This is days from diagnostic sample to the sequence report being returned, and you can see that the proportion of sequencing reports that were found to be useful was highest with the fastest turnaround times, and that completely vindicates what we thought. Fast sequencing turnaround times is necessary if you're going to find this sort of thing useful. Just want to briefly mention the other things that we got out of the sequencing data. We were the largest study of inpatients at the time of the Alpha variant sweeping the country, and we were able to show a significant increase in mortality in women. That's now been borne out by other studies, which contrasted with smaller studies which did not show any increase in mortality in hospitalized patients, although community studies did, of course. And similarly, the ITU admissions, again, we saw an increase in women, and of course this has been corroborated by data, particularly on pregnant women, where there has been increased hospitalization due to Alpha, and indeed since then more so due to Delta. Finally, just to wrap up, we were able to show that Alpha did not transmit in hospitals any more frequently than non-Alpha variants. You can see here that having a hospital-acquired infection was less likely overall if you had an Alpha infection. And if you look at these graphs, this is the community transmission of Alpha in London and outside London, community transmission of others. and the number of outbreaks, you can see it's not very clear, but the number of outbreaks was not greater with alpha than it was with non-alpha, and the size of outbreaks was also not. So that was also useful. What people are doing already in hospitals is containing transmission, and alpha was not able to impact on that. The qualitative studies were very useful, and what they showed was that running this study was onerous during a pandemic. The metadata were largely inputted manually. The links between diagnostic and sequencing labs were variable. Sequence quality was variable, and the engagement of the IPC's teams was variable, not surprisingly in this circumstance. The sequence reporting tool was liked by most IPC teams as simple and easy to understand, but they felt it needed to incorporate more metadata to be really useful. And the utility of the data for hospital outbreak management during the height of the pandemic was felt to be limited. Hospitals were already doing the maximum. There was little room for changing management. So our lessons learned and what we're taking forward, the sequencing and sequence reporting tool is perceived as helpful. We know from past studies that sequencing is useful for policy setting. It changed acute practice in about 10% of HOCIs. Where it did change practice, this was definitely related to faster turnaround times. Turnaround times were highly variable and rarely achieved less than 48 hours, even in the best of circumstances. The sequencing quality was variable and was not platform dependent. So there was variability between labs, and this is something that would have to be looked at if sequencing was ever implemented. And this study provides a template for testing the utility of sequencing for IPC going forward. So our plans are to develop a better sequence reporting tool that integrates the meta and the sequence data and also sequence directionality to generate a better, a tighter probability of transmission and linked cases. To tighten up standardization of sequencing lab outputs, we didn't actually look at that. We left people to their own devices. Most of the labs did very well. Some of the labs found it difficult. And to rerun the trial with labs carrying out their local sequencing and simpler case report forms. And it just remains for me to thank all the people. I'd also like particularly to thank Sharon and Ewan and co, who supported this study, and all the others who inputted into the design and execution of the study. Thanks very much. And I'm happy. I don't know if I've got time for questions.
Thanks very much, Judy. That was a great overview of really very unique study. We have one question at the moment. So what are the main lessons do you think can be learned and put into place to enable studies like HOCI to run more easily in future waves or outbreaks or pandemic settings?
I mean, I think not doing it in a pandemic is a good idea, because I just think people are overwhelmed, and it's amazing that anybody actually delivered it. I think what it highlighted was really three things. One, you need very simple, very good reporting tools that integrate everything at the point at which the report is delivered. It's no good faffing around with phylogenetic trees and other things on their own. You need the metadata and the phylogeny integrated into a report that can be understood by infection control teams. We got part of the way there, but not nearly far enough. You need really good quality sequencing. And that needs to be sort of standardized. We didn't do that, but it doesn't matter what platform you use, it's lab dependent. And you need really good links between the sequencing lab and the diagnostic lab and the IPC team. And those hospitals that had those links were better able to deliver. So there's a piece of work to be done on all of those three, but I don't feel disheartened. In fact, I feel amazingly encouraged by the fact that we got anywhere at all with this. First time, I think, it's been shown.
Thank you very much, Judy. We have to move on. I think there are a few other questions in the Q&A box. If you could have a look and answer, please. But thanks for a great talk. So we're going to move on now to our second talk, which is from Kate Cook, who is a research associate at the University of Portsmouth and part of their COG-UK team. And she's going to talk about their experience with using sequencing, looking at nosocomial spread and the effect of Pfizer vaccine in a hospital setting. Over to you, Kate.
Thank you. Just share my screen. There we go. Can you see that okay?
Yep, that's great. Thanks.
Okay, thank you. Can you see just the presentation? I've got the...
Yep, I can see the presentation and your pointer as well.
Okay, great, thank you. So as a quick introduction, my name is Kate. I'm a member of the Sequencing and Bioinformatics group at University of Portsmouth, and we have acted as the sequencing hub for many NHS sites along the south coast. And in this presentation, I'm just going to talk a bit about some of the analysis we've done of the data we've produced here in Portsmouth in collaboration with Portsmouth Hospitals University Trust, in looking at nosocomial spread within the hospital and the effects of the vaccination programme on COVID-19 cases and transmission. So the three main aims of these analyses were, firstly, to look at the data produced between October and May for the analysis of the distribution of lineages. Secondly, for any effects of vaccination on SARS-CoV-2 infection and lineage distribution. And finally, to have a look at the nosocomial transmission pathways and hospital-acquired infections. So in this plot here, you can see that throughout November and December, we had a few different lineages circulating within the hospital. And by the end of December, B.1.1.7, or the alpha variant, became dominant. And then certainly by February, we saw pretty much 100% of samples being characterized as this variant. Just a side note that I will be using the Pango lineage names throughout this presentation. We did have a slight enrichment of this brown lineage here, which was due to a possible local outbreak compared to the national distributions. Oops. That's not working. So in terms of vaccination, PHU were one of the first hospitals to start their vaccination programme. So you can see here on the right-hand side, we've got almost 6,000 healthcare workers having been vaccinated by the end of April with 1 or 2 doses. Around 60% had 2 doses. In terms of the unvaccinated healthcare workers, which was quite a small group of people, we saw a 10% prevalence of COVID-19 infection. And then amongst the vaccinated healthcare workers, we can see that this prevalence was 3.8% after the first dose, so very soon after the first dose. That decreased to 1.3% more than 14 days after the first dose. And then we saw 0%, so there was only 1 case of infection 14 days after the second dose in these healthcare workers. And then in addition to that, we can see that from our sequence data, we identified all of the major circulating lineages within the hospital in the infected vaccinated healthcare workers. So there were similar distributions between this vaccinated group in plot A here and non-vaccinated people in the other 2 plots I've highlighted, apart from that enrichment of the 1 lineage there. So to look at that in a different way, we did some phylogenies. So here we've got all the cases identified within PHU during the time period. You can see that there's a relatively even distribution of healthcare workers and patients and vaccinated and unvaccinated people throughout the different lineages. So what we can draw from this is that there's not a particular lineage associated with infection in vaccinated people. And then to add another level of complexity, we've got this tree here with all the community data added in. So the purple and the blue that you can see in the tree represent community cases, and the red and the green are cases from within the hospital. So what we can see here is that certain lineages are seen throughout the local community, in fact the majority of them, but particularly B.1.1.7. And then we have a few lineages which are seen primarily in the hospital, which I'll just zoom in on here. Sorry, it takes a while to load. There we go. So you can see that these 2 lineages that I've circled, we've got a lot of cases that are healthcare workers, but not so many that have spread out into the community. So what we can draw from this is that these plots are useful in starting to understand transmission dynamics. We can sort of see the order of events because points further to the left in the tree occurred earlier than cases further to the right, but we don't have any specific sort of transmission pathways identified here. So to look at this further, we did some pairwise comparisons of likelihood for direct transmission within the hospital. So we've done this for each lineage. So in these plots, we've got all the positive cases within the hospital plotted, so the sequence plotted against each other. And the blue colouring represents no direct transmission or very unlikely direct transmission between the sequences, the paired sequences, and then red represents that direct transmission was very likely and that the sequences are linked. So if we just look a bit closer at two examples, so B117, we've got most of the cases showing this blue colour, so no direct transmission. Sorry, we do have some cases of direct transmission. There are a few red pieces on the figure. But if we compare that to B.1.36.17, which is the outbreak that I mentioned earlier, the possible outbreak, we can see that there's far more linked sequences here, indicating nosocomial spread possibly within the hospital. So these cases are linked to each other. So to define whether it was nosocomial spread or not, we would need to incorporate ward data, patient location data, which is what we did here in these clustering analyses. So these plots show very similar results to those of the previous plots, but just in a different way. So each circle represents a unique genome sequence, and the color of the circle represents the ward that the sequence was isolated from. Apart from this light brown color that you can see quite a lot in all of the plots, which represents healthcare workers. And then lines between the circles, joining them together, show that they're within the same cluster. So if we have a closer look again at those two lineages, we can see that in B117, we've got quite a mess of unrelated cases based on the clustering analyses. Now, this is really interesting because obviously we had a high number of cases of B117 in the hospital, but what we're seeing here is that they appeared to be unrelated in terms of direct transmission. And this suggests several occasions of independent introduction into the hospital from the community, which makes sense because community levels of this lineage were very high at the time. And then if we look at B.1.36.17, we can see a very clear cluster in the bottom left here. So these cases that are pretty much on top of each other in the middle represent identical genome sequences. We can see that the majority of those are healthcare workers, but we've got a few from patients in different wards, which is interesting. And then this outbreak has radiated a little bit further into more healthcare workers and patients, but it's been generally pretty well contained. We've got another small cluster within this lineage, and then a range of a few healthcare worker cases that were not connected to the cluster. So this cluster represents a single outbreak. But the interesting thing here is that we can see outbreaks are not confined to wards, which is useful for hospital infection prevention and control, because they tend to manage outbreaks initially on a ward by ward basis. But what we're seeing here is that the infection has spread very quickly to different wards within the hospital. So these figures are sort of case studies of our whole data set. So each one represents a single cluster from the previous data set that I was speaking about. Each horizontal bar represents a patient, so that's one patient there. And then the orange dot at the beginning is the admission date, and then the other interesting one is the purple dot, which is date of testing positive. So what we can see for B117 is that the majority of patients tested positive within a week, in some cases just a few days from being admitted to hospital. And this is consistent with having brought their infection in from the community due to the incubation period of the virus. And then in this lineage, we've got a lot of cases that are the complete opposite of that. So we've got a lot of cases where the date between admission and testing positive was actually more than 14 days, so more than the incubation period of the virus, showing that these are likely nosocomial transmission clusters. And another interesting thing about this plot on the right is that many of these cases occurred in this grey ward, the grey coloured ward, so the colours represent the ward movements. And this was an elderly care ward. And this is a pattern that we saw throughout a few different lineages. There were quite a lot of this sort of thing happening on elderly care wards. So to conclude, we saw that the vaccination programme was very effective in terms of reducing infection numbers in staff. And we saw that infections in vaccinated staff were not enriched for a specific lineage. We observed quite a lot of nosocomial transmission between October and January. Many of these events were on elderly care wards. So increased infection control measures were introduced following these analyses, such as increased testing. And then from February of this year onward, nosocomial transmission clusters were more rare and cases were likely to have occurred as a result of introductions from the community. So we think that the reduced number in nosocomial transmission at this time is likely a result of both the vaccination programme and the increased infection control measures that were put in place. So thank you for listening. These are my acknowledgements. So thank you to everyone involved, especially Sam Robson for producing the plots that I've spoken about today. Thank you.
Okay, thanks very much for a great talk. While people are typing their questions, we have time for a few. I just wanted to start by asking you one. I think you suggested that there were some instances where you'd picked up transmissions across wards where, you know, there seemed to be spread of the outbreak, although it was largely contained.
Yeah.
I just wanted to ask if, with your metadata, whether you got any insight into the reason for this, whether it's due to staff working across multiple locations or frequent bed movements of patients before infections picked up, which we know happens quite a lot in some hospitals.
Yeah, so that's actually going to be the next stage of our analyses. We're going to incorporate a lot more patient movement data and ward data, like in those sort of case studies that I showed right at the end, to answer some of those questions. So we don't have the answers yet, but we're going to look at whether it's, you know, a healthcare worker transmission or patients being super spreaders. So that's what we're doing next.
Great, thank you very much. Actually, I think we need to move on. So thanks again for your talk. So we have two talks now about the role of sequencing in the vaccine efficacy studies. We're going to take them, the talks are going to run back to back and we'll have questions at the end. So it gives me pleasure to introduce Dr. Tanya Golubchik, initially from the University of Oxford, who's going to talk about the role of sequencing in the CHAdOx Oxford vaccine studies. Over to you, Tanya.
Thanks. Just going to try and share my screen. Oops. Look. Okay. Right. How's that?
That's great.
Okay?
Yep.
Okay, so I'm going to try and keep it quite brief because there's a very full agenda, but thank you very much to the organisers for inviting me to speak. I'm going to talk about the role of genome sequencing in the ChAdOx1 or the Oxford vaccine trial, particularly in the UK. And I'm speaking on behalf of my colleagues at the Big Data Institute and also at the Oxford Vaccine Group. Right, so just before I talk about the trial, I want to just quickly summarize our sequencing here in Oxford. We're part of COG-UK. We have been part of COG-UK from the beginning. We are a reasonably small, but nonetheless high throughput sequencing site. At the height of the pandemic so far, we've been sequencing up to about 1,500 genomes per week. This is done in collaboration with the Wellcome Centre for Human Genetics, and the analysis is done at the Big Data Institute, where I'm based, at least virtually. So from March until June last year, we were focusing primarily on sequencing samples collected from symptomatic patients and staff within local hospitals, the Oxford University hospitals and Basingstoke, North Hampshire hospitals, who've been collaborators with COG-UK. And we were also part of some healthcare work surveillance studies within the hospitals. From July onward, we will focus primarily on lighthouse samples, samples collected within Pillar 2 and processed within the Sanger Institute for cherry picking and then sent to us in Oxford for sequencing. Then from September onwards, we were sequencing a lot of samples from the vaccine trials and also for the ONS COVID household surveillance study, which is going to be talked about separately later on today. So the samples I'm going to be talking about are from the vaccine trials today. And I just wanted to highlight that the method that we use for sequencing in Oxford might be a little bit different from what a lot of the other sites within COG have been primarily using. So the method we use is called VSeq. It's a quantitative sequencing method that's based on target capture. The targets we have are probes designed against the SARS-CoV-2 reference sequence. We use these for capture and then carry on sequencing primarily on Illumina. We use the NovaSeq platform for sequencing for the most part, which is very high throughput. The method's been validated for quite a number of viruses now, including viruses that cause chronic infections such as HIV, for which we do quite a lot of work with Christoph Fraser's group in Oxford, and also viruses that cause acute infection, including respiratory infection. We've done a lot of work with RSV recently, and of course with SARS-CoV-2. The way that the method works means that the number of reads that we obtain is proportional to the input, which effectively means we can perform a viral load quantification directly on the sequencing libraries. Some of this work's been published, and this is just a plot from our publication showing that the relationship with the number of reads is maintained. So the number of mapped reads is log linearly proportional to the copies per reaction. So the trial itself is so COG-002 is a single blind multi-center randomized phase 2/3 trial assessing safety and efficacy of the Oxford AstraZeneca vaccine in the UK. It's spread across 19 study sites in England, Wales, and Scotland, and there's about 8,500 participants in the efficacy cohorts. The enrollment primarily targeted high-risk individuals, so individuals who are in occupations that are high risk for SARS-CoV-2 exposure, so in health and social care settings. That's very important because, of course, it prioritized healthcare workers who might benefit from the vaccine as early as possible, and also because it ensured that the study would be sufficiently powered to deliver the efficacy analysis that was needed. Asymptomatic swabs were collected weekly, and these are healthcare workers, so they know how to swab themselves, and they're very good about returning swabs every week. These swabs were processed through the normal Pillar 2 route and collected by COG-UK, cherry-picked at the Sanger, generally speaking, and some have been sequenced at the Sanger and also at other sites, and a large number were also sequenced within Oxford. Symptomatic cases were reviewed separately by an adjudicating panel. They were sequenced within Pillar 2, which was a more distributed sequencing process. That meant that a lot of people within COG-UK had to go and collect samples, pick them out of freezers. Sometimes there's a huge amount of work going on. And I really, at this point, would like to thank everybody within COG-UK and within the study sites who just put such a tremendous amount of effort collecting these samples, which was really key to being able to answer some really important questions quite early on. The efficacy analysis concerned samples starting from October and going through until mid-January, which is when the report was due, and that's been published. So broadly speaking, we have results from the ONS for the same time period showing that there's an increase in overall COVID-19 positivity rates in the general population, and that's actually mirrored by the percentage positive based on swab returns within the trial. And it's probably worth noting that these numbers are very similar, and that in a way indicates that the cohort that is involved in the trial are people who are extremely highly exposed and also very frequently exposed to the virus. Just to cast your mind back to what things looked like back in late 2020, early 2021, we had the decline of the so-called summer holiday variant B.1.177 and the very rapid and at the time very frightening rise of B.1.1.7, also known as alpha, and I'm just going to stick with the Pango lineage for now. And that's actually mirrored within the trial. So again, here's the UK data from COG-UK. And here is the data just from the trial, which we were able to genotype because the COG-UK sequencing was efficient enough that we were able to do this in time. These are samples that are B.1.1.7 in this kind of reddish color, and in blue underneath, we have the other lineages, including the summer holiday variant, declining in proportion. And this is very much following on what was going on with the UK at the time. And that's also reflected in the tree. So we were able to just create a phylogeny of the sequences that arose from the trial. And we can see that within that, if we overlay, so first of all the lineages, as I mentioned, and as Kate actually said in the preceding talk, the lineages are exactly the same as what you'd observe for the same time period in the UK in general. And that's reflected in the proportion of lineages and also the kind of variants that were being seen. And then if we overlay the vaccine samples, so samples from vaccinated individuals in green with the controls in purple, we can see that they're intercalated within the lineages and there's no particular preference for one over another. So overall, we were not finding any kind of funny diversity. And just to sum up, because we were able to do the sequencing in time and because of the tremendous effort that COG-UK sequencing sites have put into collecting those samples and prioritizing them, this is really a consortium-wide effort, and it can't be understated just how many people were involved. We were able to do very rapid genotyping of these lineages. We were able to validate and quality control the genome, select the ones that were possible false positives, which is also important for the trial. And in order to do that, we were then able to perform a proper assessment of vaccine efficacy by variant. That meant we were powered enough to be able to say that efficacy against alpha was actually maintained. There was some signal that it was a little bit reduced, but at those kinds of numbers, we were not powered to say that it really was reduced, but at the very least, we were able to confirm that efficacy was maintained. We didn't see any unusual diversity patterns, and we didn't see any mutations of concern, which at the time, particularly E484K, was of particular worry on the background of B.1.1.7, and we didn't see this in the trial at all. And there's ongoing work on the data set and also on more recent data as well to look at vaccine effects on viral load, on mutational burden, and so on. So with that, I would like to thank everybody who's involved and also to thank the funders. And very happy to take questions if there's any time.
Thanks very much, Tanya. That's great. We're going to move on straight to the next talk, but if you could hang around, please, we're going to try and take questions together at the end. So I'd like to invite Dr. Joy Rivers from Novavax, who's going to talk about the role of genomic sequencing in interpretation of efficacy in the Novavax studies. Thanks very much, Joy.
Hello. I seem to be having some issues with my camera, unfortunately, but I will go ahead and share my screen. Can you see that? Let's see.
Not yet. I'll let you know when we can.
Okay.
Great. Yep, that looks good.
Okay, perfect. So I'm just going to talk very briefly about the contributions that sequencing made to the interpretation of vaccine efficacy in the Novavax UK phase 3 trial. And just as a disclosure, I am an employee of Novavax. So during our trial, the SARS-CoV-2 variants changed a couple of times. So when this trial started, we enrolled 15,000 approximately adults, and they were randomized 1-to-1. The primary endpoint of the initial phase was PCR confirmed symptomatic COVID-19 illness diagnosed at 7 days or later after completion of the second dose. After this initial phase, during which we were collecting endpoints from about November to mid-January, represented by this blue box here, we did a blinded crossover. So the participants that remained in the study and had not left to receive an approved vaccine in the UK were able to get the opposite treatment that they had gotten in the initial phase. And about 6,500 adults proceeded to the blinded crossover phase. The blinded crossover phase took place between April and June of this year. And of course, that corresponds to a time period where the variant was shifting again away from Alpha and towards Delta. So just due to the timing of when Novavax has started their trial, we got to see kind of the entry of Alpha and the entry of Delta as well. So what I'm going to present today is our current analysis, which is the blue box here. And soon we will be able to speak on the green box, which of course goes out to August, and will incorporate more sequencing data for our participants who acquired the Delta variant. So during the trial, because we weren't getting sequencing results in real time, we did rely in large part on the S gene target failure. These results are published in the New England Journal of Medicine, and you can see here that we reported on efficacy in both non-B117 and B117 lineages, and that efficacy was preserved in the Alpha variant, although a little bit of a drop was observed. Subsequent to this publication, we received about 157 sequencing results, which corresponded to 62 symptomatic participants and 3 samples from participants at day 0. So the structure of our trial was to test everyone at day 0 to see if they were currently infected with COVID. And then once they became symptomatic, all of our participants were instructed to perform 3 self-swabs. So that's why it's not a 1-to-1 participants to sequencing results, because our participants could have up to 4 sequencing results. So all of the samples that we identified with the S gene target failure were confirmed to be Alpha variant by sequencing, and there were no samples without the S gene target failure that were confirmed to be Alpha by sequencing. So we had 100% concordance between the data that we used in the New England Journal of Medicine article that used the S gene target failure with the results that we received from our partners at COG-UK and Oxford sequencing labs. And so that was really encouraging. Unfortunately, going forward, the Delta variant doesn't have a convenient PCR surrogate marker. And so moving into future analysis, these sequencing results are going to be really crucial to our understanding of our vaccine's efficacy against Delta, as well as any future variants, which are also unlikely to have that convenient surrogate marker. And we have received some initial sequencing results on samples that have become positive over the summer and into the fall. And so we've already been able to confirm that the incidence of Delta variant infection among the Novavax study participants is mirroring what's going on in the UK at large. So stay tuned. I'm sure there will be a publication coming that will talk about our efficacy in Delta. But that is all for now. And I would be happy to take any questions.
Great, thank you very much, Joy. While people are thinking of questions, just to draw some parallels between the two studies, Tanya, I just wondered, I think the CHAdOx follow-up, the efficacy follow-up you said finished in January. So presumably everyone was unblinded and the placebo recipients were vaccinated, but I just wondered if there were samples being collected during follow-up and whether you're doing any work with any of the probable Delta variants coming through.
Yeah, so sample collection for weekly swabs continued until the end of June, and we've been sequencing everything we can get our hands on. Sequencing has now been moved to PHE from Oxford, so we're not sequencing those swabs anymore, but there's actually very few coming through now that the study's finished. So the next challenge is really to have a nice closed data set so we can figure out exactly what we've got and have a look. We don't have a huge number of Delta cases though, which I guess is quite encouraging in a perverse sort of way. But it'll be interesting to see what we can out of those for sure. There's also probably somewhat tangential to COG-UK. There's obviously other vaccine trials going on in other countries, which also include Delta variants, and that would be interesting to look at in time.
Great, thanks very much. I don't see any questions coming through. So I think in the interests of keeping to time, we will move on. Just thanks very much to the 4 speakers, especially for keeping to time and for some great talks. And I'm going to hand over to Professor Emma Thompson from the University of Glasgow, who's going to take you through and introduce the next session. Thanks, Emma.
Thanks very much. Good afternoon and welcome to the next session. I think perhaps Sharon might want to say something before I carry on.
Yes, I could certainly do so. We were very efficient there for a moment, but thank you. First of all, Thushan, thank you very much for your great sharing, and also thank you to all of the speakers in session 1 for a really excellent set of talks. So yes, now we're pivoting into session 2. Which is looking at care home and university studies, the impact of viral genomics in different settings. And as you heard, Professor Emma Thompson from the University of Glasgow is going to be chairing this session. Thank you.
Thank you very much. And so without further ado, I'd like to invite the next speaker, the first speaker in this session on care home and university studies, Dr. Maria Krutikov from Imperial College London, who's going to give us a talk on COVID-19 in care homes. Maria, the stage is yours.
Thanks very much. Can everyone see my screen?
Yes, perfectly. Thank you.
Excellent, thank you. So thank you very much, and thank you for inviting me to speak today on behalf of the VIVALDI study. I'm Maria Krutikov, I'm an infectious diseases registrar, and I'm a Wellcome clinical PhD fellow working on the study. So in the next 15 minutes, I'll be giving you a brief overview of the study and some of the results and progress to date, and I'll highlight some of the key challenges that we need to overcome in order to use genomic sequencing data in our analyses in order to answer some of the questions that we would like to answer in the future. So this slide gives just kind of it's just showing some of the headlines relating to COVID-19 in care homes, and I'm sure everybody listening is very aware of this. But I think one of the key challenges that we faced throughout this study has been relating to the sort of lack of a research infrastructure within care homes, which has meant that we've had to kind of overcome quite a few particularly logistic issues in order to get our study up and running. But clearly there is a very urgent need for this work given the devastating impact of the pandemic in care homes. So the VIVALDI study is a prospective observational study, and it's been running since the early pandemic and is currently funded until April 2022. And it's funded by the Department of Health and Social Care of the UK government. And the aim is to investigate SARS-CoV-2 infection, immunity, and its duration in care home staff and residents. And the aim of this is to inform public health measures to reduce transmission of infection and subsequent death. So the study started initially in 100 care homes that were all belonged to the Four Seasons chain. And these were geographically distributed, but in order to improve generalizability of our findings, we expanded to a total of approximately 350 care homes that are run by a range of providers, so large and small, for-profit, not-for-profit, and independent. And these all care for older people. And we collect data from all staff and residents at these sites. So this is an overview of the study design, and it's made up of three main components. So consenting participants, so that this side on the left with the green boxes shows the blood sampling side. So consenting participants undergo blood sampling on a 2 to 4 monthly basis. And this is up to 5 times over the course of the study. And these samples are tested for nucleocapsid antibodies using the Abbott assay at the Doctors Laboratory in London. And they also undergo more detailed immunological testing of cellular and humoral responses by Professor Paul Moss's lab in Birmingham, as well as proteomics by Marcus Ralser and his team at the Crick. This middle section shows the PCR results from pillars 1 and 2 that we also collect as part of our study. So these tests are performed weekly in staff and monthly in residents. And there is also additional outbreak testing for hospital as part of Pillar 1, which is run by PHE, which is now UKHSA. And we collect this data for all staff and residents in participating care homes. And then these data are linked with vaccination records from the National Immunisation Management System, so the NIMS system, and hospitalizations from hospital episode statistics and deaths from ONS, as well as care home level data sets such as building characteristics, staff and resident turnover. And these data are all linked and pseudonymized by NHS England using a pseudo identifier, which is based on the participant's NHS number. And then we're able to access this very rich linked data set that enables us to address policy important questions in this population. So I'm now going to run through a few of the results from some of our studies and then describe some of the issues and challenges relating to enabling us to use genomic data to address our study questions. So this is a study that was published in the New England Journal, and it was looking at, it was performed just before the third national lockdown in the UK, which was in December and January 2021. And it aimed to describe the proportion of B117 variant cases in care homes over the autumn and early winter of 2020. And it used PCR results from all Pillar 2 samples that were processed in the Milton Keynes Lighthouse Laboratory and reported on the proportion of SARS-CoV-2 isolates with S gene target failure. So this is a marker of the variant, as we've already heard. And we also reported on the median cycle threshold value over the same time period, which is a marker of viral load. And we showed that over the period of October to December 2020, there was an increase from 12 to 60% of positive samples with S gene target failure and a corresponding drop in median CT, particularly in the southeast, east, and London, as shown in these figures here. And we were able to retrieve sequencing results from 2 samples in this study, and both were B117 variant. And from this was one of the first reports of B117 variant within care homes. And it also, there was a suggestion that the variant was initially more prevalent in staff than in residents, with a corresponding rise in residents towards the end of the study period, suggesting that the introduction may have been through staff. But unfortunately, we didn't have enough sort of data to be able to make some solid conclusions on that. This is a paper that was performed just after the rollout of vaccination in the UK, and our aim was to report on the risk of reinfection in care home staff and residents. So we linked the Abbott antibody results from antibody testing from just under 700 residents and 1,500 staff in the 100 care homes that we started our study in. And we linked it to pillar 1 and 2 PCR results in order to estimate the risk of infection based on nucleocapsid antibody status. And this was a marker of past infection. And so we found that previous infection reduced the risk of reinfection by approximately 85% in residents and 60% in staff over our study period. So we were the first study to report on the efficacy of 1 dose of vaccination, and we performed this study again following the widespread rollout of vaccination in care homes. And we used data from the NIMS vaccination database and pillar 1 and 2 PCR results. And we were able to estimate vaccine efficacy in just over 10,000 residents and show that this was 56% after 28 days and 62% after 35 days. And we also found that CT values were lower in unvaccinated when compared with vaccinated individuals, which suggested that there may have been an effect on of vaccination on infectiousness, and this was recently published in the Lancet ID. And so this slide just shows some of kind of the study progress to date. So we've been able to recruit just under 6,200 individuals from 220 care homes to have blood testing. And this has been with a huge effort, largely with the help of all of the care home staff and particularly the managers, and also with the clinical research network. But we're also collecting data on all staff and residents from 330 sites. And this is over 60,000, on over 60,000 participants. And this is under the COPI notice, which is in place until March 2022. We also have an existing collaboration with the UCL Bartlett of the Built Environment and with COG-UK. And we've had some substantial policy impacts as a result of our findings. So there have been some changes to visitor restrictions to care homes, the establishment of the Infection Control Fund, which has enabled care homes to fund things like sickness pay for staff, which we've previously shown to be a risk factor for infection. So sites that don't pay sickness pay to staff have higher rates of infection, and also limited cross-site working for staff. And so to date, we've been limited in the extent to which we can use sequencing data in our analyses. And this is largely because we're reliant on existing PCR sampling infrastructures, and they're predominantly from Pillar 2, which is run by the Department of Health and Social Care. And we have therefore little control over whether samples are cherry-picked and then sent on for sequencing. So this has been a very large piece of work that we are making good progress, but are still trying to unpick. And this diagram illustrates the sort of ideal scenario for the flow of samples from VIVALDI care homes to make sure that they're sequenced. And we've developed this, as I say, with colleagues within the logistics team and with the laboratory directorate and with COG-UK. And so one of the things we've done is we've developed VIVALDI barcodes. So they have a VIV prefix that are sent to all VIVALDI care homes in addition to their regular sampling kits. And these barcodes should be affixed to the kits. And we also include some pink kind of outer stickers to go onto the outside of the boxes that the kits are sent back to the labs in. And this is supposed to flag to labs that these samples need to be cherry-picked if they're positive, and then sent on for sequencing. And subsequently, the PCR results and genotyping data from the reflex assays that are now routinely performed as part of Pillar 2, as well as CT values and the sequences from COG-UK are supposed to sort of flow into through SGSS and through the CLIME database into our sort of linked VIVALDI dataset to enable us to answer some of these important questions. However, this green table shows some of the challenges that we face and some of the ways we're trying to overcome them. So the first reason is that the barcodes aren't applied. The kits don't come with VIVALDI barcodes. And so we've just undertaken a telephone survey with care homes that are taking part in our study to try and work out some of the reasons for this. And some of them include that the care homes are using up old stock. They lack of awareness about the fact that these stickers need to be applied, and use of postal return kits when staff are using lateral flow tests. So we're planning a webinar with these care homes in order to talk to them about some of the issues and provide the information that they need. Another issue is that the samples aren't cherry-picked by the Pillar 2 labs and sent on for sequencing. And so to address some of these issues, our colleagues in the Department of Health are also changing the appearance of the swabs so that they can be flagged when they're booked in because they look completely different, and also this pink outer sticker that I described earlier. However, as the labs are really busy and have limited capacity, and because of differences in practices across different labs, most of our samples are still not reaching the Sanger for sequencing. So we are in ongoing talks with the lab directorate about how to address this because our study remains prioritised for both cherry picking and sequencing. So as I previously touched on, the sequencing data would really enhance our existing data set and allow us to address some of the current questions about SARS-CoV-2 in care homes. And these include the characteristics of vaccine breakthrough infections, the risk factors for care home outbreaks, how different variants influence this, and the main routes of transmission into the homes. And this is something we haven't fully been able to address so far, because we need good coverage of sequencing, and we've not managed to achieve this. So this has been highlighted as a research priority in recent reviews, and we are working on it. And one of the other things we are currently considering is longer term funding opportunities in order to create a care home platform for research. So thank you very much for your attention today, and I'd really like to thank all of the study participants and the care homes, as well as the rest of the study team and our collaborators. Thank you.
Thank you very much, Maria, for a fantastic talk and a huge and really impactful study for describing that so well. We do have one or two questions. I think you've probably actually addressed one of them already at the end of your talk. And what I think is that it's quite a general question as well and might be better applied at the end. So I think what we'll do is maybe take questions at the end of this session. So there's another talk, and I imagine the same question will be important for the next speaker as well. So if you can hang on, that would be great. And we'll bring on the next speaker and then come back to the questions after that. That's okay.
Thank you.
So thank you very much, Maria, and we'll come back to you shortly. The next speaker is Dr. Dinesh Aggawal from the University of Cambridge, who's going to talk about the genetic epidemiology of SARS-CoV-2 in universities, or in one university. Dinesh. Thank you very much. We can see your slides. That looks good. You probably just need to put it up. That's it.
Brilliant. Thank you. Thanks for having me today. I'm Dinesh. I'm a Wellcome Clinical PhD Fellow as well, and an ID Registrar at the University of Cambridge. And I'll be talking through the use of sequencing to understand the dynamics of transmission at the University of Cambridge. So for those not aware, at the University of Cambridge, we've got two routes for testing for students and staff. So students within college accommodation have access to an asymptomatic screening programme, and all staff and students have access to a dedicated symptomatic pathway testing programme, as well as access to test and trace. Just a quick outline of what the pooled screening entails. Essentially, individuals living in college accommodation, which is essentially synonymous with student halls, can pool their swabs up to 10 individuals in a household, which goes on to be tested. And if positive, the students are then offered or then take a individual test to confirm their individual test was in fact positive. So this was essentially employed to increase case ascertainment and to keep tabs on what the infection rates within the university were during the terms last year, and that's continued this year as well. So through both pathways, we confirmed 788 cases in the first term of last year. And then we had an additional 184 cases confirmed through test and trace. So we used this kind of high case ascertainment to answer three main questions with genomic epidemiology. One, what the kind of source or drivers for transmission were within the university, what methods we could use to mitigate transmission, and also whether there was any level of transmission between the student and local communities. So as a result of these kind of tests that I just described, we're able to include 482 high quality sequences from university members in this study, which were comprised of just over 450 students and 30 staff. And coincidentally, we also had 972 cases from the COG-UK dataset representing Cambridgeshire, which represented just under 750 of the general public, just over 120 healthcare workers and just over 100 patients. So on the left here, we can see a maximum likelihood tree of the entire study cohort. And the first thing to notice is the segregation of the community and university students, with a particularly large cluster of university individuals seen at the top left, called cluster 1. On the right, we can see the lineages in the university on the top right and the community on the bottom right, with little overlap between the lineages and the two populations. And these are Pango lineages, just for reference. To understand our cohort in a bit more, with a bit more granularity, we use CIVIT to identify transmission clusters. And the entire cohort was broken down into 198 possible transmission clusters. Interestingly, just over 90% were represented, 90% of cases in the university were represented by just 8 clusters. And the largest transmission cluster, cluster 1, which I identified earlier on the maximum likelihood tree, that represented just under 70% of the university cases. And interestingly, it was epidemiologically highly diverse, with over 200 households represented in 29 colleges and 28 undergraduate courses. So the first thing to understand with this particular cluster, given it represented so much of the university, so many of the university cases, was to see if it was introduced kind of multiple times into the university, or if it was truly consistent with one single outbreak. So to do this, we first of all looked at the introduction of the cluster into the university cohort as compared to the national COG-UK dataset, and we found that it essentially was first sampled in the UK in the first week of October, just prior to the university term starting. And then by the second week of its presence in the UK, it was found in the university, then predominantly found within the university, and not really in many other regions. So it seemed like it had been introduced, but also we evaluated this further using a timescaled beast tree, as you can see on the right, and demonstrated that this particular cluster was highly divergent from any local Cambridgeshire clusters in the surrounding region, with the time of most common recent ancestor dating back to early May. Finally, not shown here, we also used A to B COVID, which demonstrated this was consistent with one single outbreak. So to utilize this large cluster to understand the drivers for transmission, we first of all wanted to understand how this particular cluster came about within the university, or at least spread and dispersed amongst the university students. And we found that by linking it with contact tracing data from test and trace, a particular nightclub was identified where individuals early on in this cluster seemed to congregate, and that implied it was probably the source where this particular cluster became then present in multiple colleges and courses. We also employed a BD skyline model to understand the transmission dynamics of this particular cluster throughout the university term. As you can see on the bottom left-hand corner, there was a rise in the effective reproduction number at the beginning of term, which settled as university infection prevention control measures were put in place. But then there was a subsequent rise just before the national lockdown, coinciding with the announcement of national lockdown in the November of 2020. Once again, the students who were positive in this cluster and also around this time happened to report a common exposure, or the predominant common exposures that they reported were once again linked to nightclub venues. Reassuringly, you do see a sharp decline in the effective reproduction number after national lockdown was put in place, with identifying compliance of the students with this policy. We also looked at other settings in which transmission was occurring within the university, and unsurprisingly, we found lots of transmission within student households. In cluster 1 in particular, 50% of students had a sequence identical to at least one other student living in the same or a neighbouring household. Though we did see transmission within households, we also saw this as an opportunity to mitigate transmission, and we found several instances where, when students were identified to be infected within a household and infection prevention control measures could then be targeted to them, the infection could then be mitigated to that particular household. And the largest example of this is seen in the top right-hand corner, which is cluster 2. We also found a household attack rate within our student household of 7.8%, which is lower than other similar settings reported elsewhere. We found evidence of transmission between students in the same course and year of study. And in cluster 1, once again, we found just over 60% of students had an identical isolate to at least one other student studying in the same course in the same year. And using a simulated model of expected infections amongst the different years of study, we found a highly disproportionate number of infections within the first year of students. We also found minimal evidence of staff-to-staff or staff-to-student transmission, which was reassuring for our members of staff. And finally, to identify transmission between the university and its local community, we found phylogenetic segregation of the majority of community and university students, as I demonstrated earlier. We also found transmission clusters of less than 15% contained both university and community cases. And the largest one, which I've discussed earlier, cluster 1, was only comprised of 1.8% of community cases in total. Linking the entire data set to the national contact tracing data, these findings were corroborated by epidemiological data, with only one community individual sharing a common exposure event with a university student at a social venue that happened to also be part of the same genomic cluster. Contrastingly, we found 19 epidemiologically linked clusters were refuted by phylogenetic variation. One thing we did highlight was we did find multiple links between clinical medical students and hospital cases, highlighting this particular cohort as a possible route of transmission between the university and its student and its local population. So in conclusion, we found the majority of university cases were linked to a single genetic cluster, with the nightclub as a likely source and also holding a critical role in propagating transmission of this particular cluster. We found evidence supporting transmission within households, courses and year groups, and these could be targets for intervention. But also we found significant evidence of success of the university screening programme and other control measures, particularly outbreaks being controlled to single accommodation blocks, the majority of transmission either occurring within a university or within the local community, and also compliance of the students with national lockdown. And as I mentioned, we found evidence suggesting links between medical students and hospital staff and patients, which would perhaps highlight them as a cohort that require vaccination. Perhaps not an issue in the UK, but definitely one globally. I'd like to thank everyone who has been involved in this study, which kind of has been a huge operational program, not just from COG-UK, but also the screening program. And thank you all for listening to me.
Thank you very much, Dinesh, for an excellent talk, very interesting. Just a quick technical question from James Noakes here. I may have missed this, but how are you defining a transmission cluster?
Oh, sorry. Yes, I might not have mentioned that. So transmission clusters were defined with CIVET using default settings. So essentially where one case is within 2 phylogenetic neighbors of another case. So we kind of used a more longitudinal transmission cluster definition to represent likely transmission within a well-sampled cohort rather than just something like SNP distance.
Okay, thank you. And there's another question here for you, and then I might come back to Maria just about interventions and you know, how we can use sequencing, for example, to intervene. Do you think the segregation of community and university cases is more to do with how institutionalized students are in Cambridge? Would we see this in another city-based university?
Yes, so I mean that
are intermixed a bit more in private accommodation and so on.
Yeah, absolutely. So so obviously the University of Cambridge has a kind of very well-defined structure, and there's not just of the university and the community, but also within the university itself. So that no doubt biases the findings. The only thing I can say is that this is the only study so far that's kind of fully reported of genomic epidemiology within a university setting, and kind of out to review currently for publication, but we'll see. But essentially, yes, whether the findings are completely translatable elsewhere is a question that would be helped with similar studies being set up elsewhere to address that.
Okay, thank you very much. And Maria, I was going to ask you at the end of your talk about, in fact, I'm going to merge a couple of questions here. So there's interest in which policy changes you think might have been most effective in nursing homes, and also, have you been able to go back and look at the effect of these interventions? Do you think care home staff are behaving differently now, for example, than they were at the beginning, and how can you monitor that?
Thank you. That's a really interesting question, and I think it's hard to answer succinctly, but essentially I think some of the main interventions that we think were the most effective were the introduction of regular testing for both staff and residents. And that was first introduced in June 2020, and also the widespread vaccination campaign was clearly very effective and was very quickly carried out across all care homes. All of the other interventions in terms of cross-site working, sickness pay, visitor restrictions on visitors, I think it's anecdotally they have had an effect. I think we are trying to come up with analyses to try and measure the effect of these interventions, but given that they occurred at different periods of the pandemic when the background incidence of infection also rose or dropped, it's really hard to directly measure the effect. And I think that if we were able to get good coverage of sequencing data, even from just 1 or 2 care homes, which is I think a strategy we're considering now, given the difficulty we've had with the scale of the study, then we could at least make some inferences about transmission routes and work out whether we think it's coming in from visitors or staff or hospital, or and that would be incredibly helpful, because we could combine that with all of the epidemiological data that we already have about staff and residents. But I think it's a really interesting question.
Yeah, and you said that that you thought that a lot of the transmission was probably staff to residents, which makes sense. Did you get the feeling that that was asymptomatic mostly, or that people were going to work because they needed to be paid, as you had said, in terms of policy?
So I guess it depends on the point of the pandemic. Once regular screening was introduced, staff were being swabbed every week, regardless of symptoms. So I think it's unlikely that symptomatic staff were able to come into work, and also asymptomatic staff would probably be detected quite early. I think that the point about the sickness pay is that, yeah, if someone has symptoms and isn't going to get paid, then they might potentially go to work, especially if they have kind of soft symptoms. But I think that's one of the key things in terms of the regular screening. That's why it's so important, to continue that.
Yeah. Okay, lovely. Well, thank you very much to both speakers. We are finished and on time, so thank you. There was another question which I think was probably more of a comment about, and Maria, I think you've obviously gone to great lengths to try to get your sequences identified for sequencing, and it's a problem everywhere across the whole consortium. I think we're trying to get samples from Scotland as well dug out for us that we think are of public health importance, and it's quite challenging, and maybe addressing that is something that we should all be thinking about a bit more in the future to try to enable great studies like this. Okay, thank you very much. There's a very short break now, and we're coming back at 2:30, so you've got about 4 minutes, and see you shortly for the next session. Thank you. Thank you again to the speakers.
Following that short break, we're going on to our third and last session today, which is really focusing on public health and national infection surveys, looking at the role played by genome sequencing of the virus. And so the chair for this session is Dr. Theo Sanderson, who works at the Wellcome Sanger Institute and the Francis Crick Institute. So Theo, thank you for chairing, and if I can hand over to you, please.
In the UK, we've had the luxury of 2 different surveys that survey the community for infections, and another one is the REACT survey. So I'm delighted to introduce Oliver Eales from Imperial College, who's going to tell us about sequencing work in support of that. Oliver.
Thank you. Let me just share screen. Can you see this, Theo?
We can. Take it away.
Thank you. Yes, I'm Oliver Eales. I'm a PhD student with Stephen Riley at Imperial College London, and I've been working on the REACT-1 study, and so I'm just going to present some of the results we've got so far. It's not yet peer-reviewed as well, so let's just move on. So for those of you who don't know what the REACT-1 study is, it stands for Real-Time Assessment of Community Transmission, although that is rather forced. And it provides a 2-week snapshot of the spread and prevalence of COVID-19 across England. How it's done is each month we send around 90,000 to 180,000 swabs to random individuals across the country, who then complete these swabs. They go off to a lab and we find out if they're positive or negative. And then we can do lots of stuff with it, like estimating reproduction number over those 2-week snapshots. And looking at how odds of infection varies with different socio-demographic characteristics. But what I'm going to be talking a bit about today is the genomic sequencing that's being performed by Consortium Institute for the REACT-1 study. So what happens is positive swabs where there's a high enough volume and it's likely that a lineage can be determined with the sample. They get sent off to the Consortium Institute. They then perform genomic sequencing, and then we analyze to see how stuff is sort of changing over time and normal other things. We first started doing this in round 8, which was in January 2021. We're now at round 14, which is just finished and will be out sometime next week. But I'll be talking about round 8 to round 13, which is from January until July. Obviously, most of this is quite well known, but SARS-CoV-2, there's a lot of mutations, as all the other talks have shown. And some of these are just neutral, but some variants of concern have started popping up all around the world. Just for nomenclature, B117, which is in all the figures, is the alpha. B1351 is beta, and then the B16172 is the Delta variant. There's also other variants under investigation. Obviously, COG-UK has done absolutely tons of genomic surveillance and probably has the biggest data sets of across the world. So what REACT-1 kind of does is it's more complementary because we get much smaller sample sizes. So though REACT-1, because of its sort of random nature, is very unbiased, similar to the ONS study, we do get much smaller sample sizes, so we can't always do the most powerful analysis with it. But it acts as a sort of check for against sort of estimates that might be made with, say, more biased samples, due to, say, like surge testing and other such things. But here's our prevalence over time and the number of samples where we've been able to determine the lineage. So in January, around 8, we were able to determine about 1,000 because prevalence was so high that we detected a lot of positives. But by, say, late April, when prevalence was at its lowest, we only managed to determine the lineage of 26 infections. So first focusing on January to March of 2021, which is when the Alpha variant was circulating, we found a large degree of diversity in the lineages circulating in January, but this decreased massively into February, which is when prevalence was decreasing and probably a lot of lineages were going extinct or locally extinct. And by round 10, which was March, we were only detecting the Alpha variant. So the proportion of Alpha in January was at 83%, and by round 10, it was at 100%. And using some simple regression, we can estimate a daily growth rate difference between Alpha and sort of all the other strains that were circulating, which we refer to as wild type. And this corresponds to an additive R advantage of 0.31. If you prefer the multiplicative one, it's about 35% more transmissible. Spatially, we found that the Alpha variant was highest in London and other regions in the south of England, which is not very surprising, seeing as it was first detected in around Kent. It was much lower, though, in areas like Yorkshire, where in January it was only at 57%. However, by round 9, which was in February, basically all the spatial heterogeneity had disappeared. And I think you can see that more clearly in this plot, where basically the more yellow an area is, the higher the proportion of the Alpha variant. And as you can see, in round 8, it was much higher in the south towards Kent and London. And then that basically disappeared in round 9 when most of the samples were the Alpha variant. We were able to look in at just round 8 due to sample size issues, but in round 8, we found there was an increased odds of infection in 18 to 54-year-olds. And we thought this might be due to regional effects, but doing a combined model, we found sort of no difference in the distribution. This is particularly interesting as around November 2020, other studies found that there was a high proportion of Alpha infections in school-aged children. So the fact that it had actually kind of reversed the pattern by this time suggests there was some sort of change in the advantage of Alpha over wild type in age groups over this time, potentially due to school closures over Christmas and into January. We were able to look at the sort of proxy for the viral load. So the N gene and E gene CTE values, which is what we measure in the REACT-1 study. We found that for Alpha, it was about 1 lower than the wild type. The exact numbers are there, 1.33 for N gene and 0.9 for E gene. This approximately corresponds to a viral load that's 2 times higher, which could explain the sort of increased transmissibility that we observed. It could also just be that the increased transmissibility causes us to detect an N gene and E gene that's higher or lower, sorry, just due to the sampling of our study. So we can't really isolate the effect properly. We also get reports of any symptoms that someone exhibits, and we find that there's actually a similar proportion of individuals exhibit the sort of classic COVID-19 symptoms, which are loss or change of sense of taste and smell, persistent cough and fever. However, there's a slightly higher rate of any symptoms, which includes a big list of like 30 different symptoms in the people infected with the Alpha variant. although it's not actually significant. What a significant result we do find is that people who were infected with the alpha variant are actually more likely to be test positive for antibodies. So in round 8, we sent a subset of our positive participants a lateral flow immunoassay, which tests for antibodies. And we find a higher proportion of the alpha infections were testing positive for antibodies. So about 83% compared to 73%. We thought this might be explained by the sort of higher viral load in alpha infections, or maybe by the age distribution. So we did some sort of regression modeling. And as you can see from this graph, looking at the green dots with the line, if you include sort of other covariates in the model, there's very little change to the actual results. There's still an increased odds of testing positive for antibodies if you've been infected with alpha versus wild type. And including any number of those covariates doesn't change anything in a material way. In January, we detected some other variants of concern and variants under investigation. The most notable is probably that we detected 4 beta infections, sort of around London and the sort of areas around London, so the southeast and east of England. Though this doesn't seem like a lot, it's only 4, because of the way our sampling is done, and we can actually put a sort of estimate of the number of people infected at any one time during that period. And just so a single detection corresponds to about 800 infections in the community. So for beta, we were actually estimating about 3,000 people were infected with it at the time. And the lower bound, more importantly, is actually over 100. And beta, I think it was over 600 or so people they were estimating as being infected with beta. Obviously, beta hasn't really taken off in England. And so in round 10 and 9, we didn't detect any beta, and we were able to put an upper bound on the sort of growth rate advantage of beta over alpha. And we found a 98% probability that the growth rate of beta was lower than the growth rate of alpha. And what we kind of think explains why some of these variants of concern and variants of investigation got to high enough values in the community in January, but disappeared in the following 2 weeks, is that the number of people traveling abroad in January, which is round 8 here, was much higher than in the subsequent rounds. So there was still much more of a chance for these variants to get into the country. And as you can see there, there's a lot more travel to London, which could explain the geographic distribution of those beta variants and some of the other variants under investigation that we looked at. And here's just another graph of the proportion of people who've been abroad in the last 2 weeks. And as you can see in round 8, it's mostly people just traveling into central London, which is, I think, also where a lot of the variants of concern were detected in the sort of wider COG-UK data. Moving on to April to July, which is our rounds 11 to 13, we started detecting the Delta variant. And in round 11, which was our smallest sample size, we only detected 7.7% of lineages as being Delta, the rest being Alpha. But a month later, it had increased to 78.3%. And then finally, another month later, it was at 100%. Rather unfortunately, the 2 rounds in which there were both Alpha and Delta detected in our study also had the smallest sample sizes, so we couldn't actually do any comparison of them for that. And then round 13, when we were back to a big sample size, only had Delta, so we weren't able to do any comparisons. But we can estimate the sort of daily growth rate difference between Delta and Alpha. And we end up getting this additive R of 0.86, which roughly corresponds to, I think, Delta being 2 times more transmissible than Alpha, which is in line with some of the other estimates that are out there. So just a quick conclusion. React-1 study design leads to a collection of unbiased samples for analysis, although because we're testing so many people just hoping to get positive results, we usually get very small sample sizes. So it's much more useful as a sort of complementary check to see if other estimates are consistent with our estimates. Collection is still ongoing. We're just finishing round 14, and the results will be public sometime in the next 2 weeks. And hopefully they'll be useful in the next 2 rounds, especially at looking at how the Delta sublineages vary over time and sort of isolating any differences that might exist there. And finally, I'd just like to thank everyone involved in the study. So everyone at Imperial College London who's helped with the analysis, Ipsos Mori, who's collected a lot of the data for us, and then the Quadram Institute, where they've actually been doing the genomic sequencing for us and putting up with my emails with incessant questions about it. And then also a special thanks to everyone who's actually participated in the study. I think we're up to around 2 million people who've participated now. So maybe some of you on the call have actually provided a swab. So thank you very much for that. That's it. So maybe some questions or.
Thank you very much, Oliver. I think we have time for one very quick question. So that is from Zoe. She's asking about a situation in which there's limited capacity to sequence and how you would prioritize that. I'm guessing you actually probably sequence as much as you can, but also I guess the question is, how do you choose which areas to sample from?
That's, good question. So the actual large study, we sample at the sort of LTLA level. So England split up into around 315, and we get around 200 people to participate. And they're sort of randomly in from each LTLA, and they're sort of randomly invited using the NHS GP list. We'll then get all of our positive samples, but that will be a much smaller subset. So I think our biggest round was round 8, and that we had maybe 2,000 positives. The reason we then basically sample all of them, and there usually isn't, because they're such small sample sizes, usually there isn't much concern about which ones to sample. It's usually just which ones have a low enough N-gene CT value. So I think our cutoff at the moment is around 34, but it was 32. And that's just because anything higher, it's very unlikely that we get a result from the sequencing. And then just some other factors like if there's enough volume in the sample.
Fantastic, thanks so much. I think there's some more questions in the text which you might be able to get to, but thank you very much, Oliver. I'm going to hand back to Sharon to close us out.
Thanks very much, Theo, for chairing that last session. So it's down to me to say the really important thank yous to everyone today, starting with our speakers and chairs for contributing to such a really strong showcase. It's been absolutely brilliant, so thank you so much. And I'd actually also like to thank them for their contribution to the pandemic response and for working with us at COG-UK. This makes a difference to the way we're handling the pandemic in this country. Special thanks go to Elaine Westwick, Andy Jeremy and Kim Smith, who actually organised this meeting, and it wouldn't be possible without their help. Thank you to the audience for coming and contributing your questions. So we look forward to welcoming you back to our next showcase event, which is going to be now in 2022. So thanks very much for coming, and I hope you have a pleasant afternoon. Goodbye.